Hello there, and hope that everything is going well for you. I know this video is coming out here at the last minute, and just thought I'd go ahead here and take you through a couple of questions, kind of show you how the first exam is written. So first thing, just to kind of show you, I happen to have the practice questions up here on my screen. Hopefully you guys can see it. And then the second thing I happen to have are the objectives for exam number 1. So a couple of things again with the objectives. The objectives are what I make the PowerPoint from, and also the other thing too, they're also what I base the exam questions off from. So some students will go ahead and they'll say things like, oh my gosh, like even though there's a study guide and everything else, like that test just, it knocked me out. Or again, they just had difficulties, and then they go ahead and they ask, like, where did this stuff come from? I wasn't ready for it. And, sometimes there's a differencing opinion. I feel that, here's the objectives, here's what, again, the lectures are based off from. And that's why I made the test questions as a sample, just kind of show you, kind of walk you through things. So first off, number 1, when I do write the exam, I'm going to keep things pretty much in sequence. And so what I mean by being pretty much in sequence is that the idea of what you see here, like for the scope and history of microbiology, for the most part, those questions will be in the very beginning. And then I'll transition over to the second set of objectives and then into the third. My reason for that is, honestly, I really don't want you jumping around. I don't want you going from a question dealing with taxonomy, dealing with cell structure, dealing then with disinfectants, and then going over to media, and then back into the first set of objectives. I want you thinking about what's going on and looking at specific content areas, okay? So just as an FYI, I want you focused, all right? So that's in general what I'll do with the structure of the exam. Now, taking a look at, again, the objectives. Some of these objectives are going to be very straightforward, and they're just going to be kind of a rote memory. And then after that, the other ones, again, are going to be more application. In here, I showed you more of the application, so that way I could go ahead and give you an idea about what's actually going to be going on, okay, and how to hit the application questions, because obviously they're much more difficult. So with that, going ahead and taking a look here at what we happen to have, question number 1 is pure rote memory, okay? The question is, identify the major contributions discovered by Pasteur. So you happen to have number 1, germ theory of disease, number 2, pasteurization, 3, fermentation, Pasteur's postulates, and then he has, of course, disproved abiogenesis, also known as spontaneous generation. And if you can kind of notice the format right here, I happen to have 5 choices, and then I happen to have the choices down here as the multiple choice answers. And you'll notice that I don't have necessarily any of the all of the above or none of the above. So I'm actually making you go through these. So first off, number 1, where do I get this objective from? So I'm going to the exam number 1 objectives, and boom, it's right here, right? List at least 3 contributions of Louis Pasteur to microbiology. And if you go back to the first video, you'll see the contributions there. Now, obviously, you guys are taking this test online. I really think that if you're taking it on paper, it'd probably be a little bit easier because you could use some test taking strategies. So with that, I don't know how much highlighting you can do on your end on Canvas, but, taking a look here, first off, number 1, did Pasteur come up with the germ theory of disease? The answer for that, of course, would be no, he did not go ahead and do that, okay? The other part here, when you start taking a look at what Pasteur also ended up going ahead and not doing, he did not come up with Pasteur's postulates. If you know that one right there, that happens, of course, to be Koch's postulates. So that is not something that Pasteur is credited with. That's a distractor, okay? So really, pasteurization, fermentation, and then he disproved abiogenesis, also known again as spontaneous generation. So, if I had the ability here to highlight these two, they happen to be wrong. And so anything with the number 1 and the number 4 here happen to be wrong. So if you notice, I could cross off letter A as a choice right there. I could also cross off letter B, and I can also cross off letter C, and I can also go ahead here and cross off letter D. All of those choices are wrong because, again, they contain either Roman numeral 1 or Roman numeral number 4. And so taking a look, I happen to have 2, pasteurization, 3, fermentation. Number 5, he disproved abiogenesis. So if you notice right here, the correct answer happens to be letter E. Okay. Now, the next one, okay, goes through, again, the kingdoms. And taking a look here at the kingdoms, all right, so I'm going back into the objectives here. And I went through, and again, I talked about, some of the characteristics of, the kingdoms. What is going to make a bacteria bacteria? Talked here again about the Woos system right here. Talked about what made those unique right there. Talked again about what happens here with that. The classification of archaea from eubacteria and everything else. So went through those, okay? So taking a look at this one right here, you recently discovered a prokaryotic organism. So back to the kingdoms. Remember how we break things down, eukaryotic, prokaryotic. Well, this thing is prokaryotic. You have decided to classify the specimen into the genus Rixidia. I am going to tell you, if you hit anything at all where you're going, oh my gosh, we never talked about this species, we never talked about this thing in class, don't worry about it. And the reason why you don't want to worry about it is this is an application question. I don't expect you to know this at all genus of organism. I'm just putting it out here, okay? So with that, going forward, all right, I don't want you panicking about that. Know what the question is, and I still haven't even hit it, okay? So, moving forward, scientifically speaking, these organisms are not a single coherent group and are classified into separate species. Even though the organism does not belong to a coherent group, it does share the same characteristic that all Rixidia exhibit, and that is a minimal amount of peptoglycan with a solid layer of lipopolysaccharide. What kingdom would this organism be classified into? I know I read the paragraph for you, but I read the paragraph to get to the question. That's the important thing I'm going to be doing here for you. The important thing is the question. The question is asking you, hey, what kingdom is this thing going to belong into? So taking a look at it, again, prokaryotic. Well, if you're looking at prokaryotic, you're now only looking at 2 kingdoms. So again, you could cross out the animal kingdom. You could also cross out Protista, plants, and you could also, again, cut out the fungi. So notice there how I have them highlighted because I'm crossing them out. So now I'm down to 2 choices because these are the only 2 prokaryotic kingdoms. And then after that, the next thing happens to be, again, this important idea that they do have peptoglycan. And so taking a look, if you do have peptoglycan, that's going to be the eubacteria, the true bacterial kingdom. So that's going to be number 2, or I should say letter C. Question is number 2. My mistake there. All right. All right. Number 3. Here is another question, right? And this one again goes into the domains. And so it goes back into this whole idea here of classification. So I've taken this domain stuff by Woos, and again with it, taking a look at number 3 and pretty much taking 3 and 5 and putting them together. And this is a real life situation here. And what I mean by being real life situation, I actually went to the University of Google, as many individuals like to go ahead and say that they got their education from Google. Also, this is a reason too why you should not depend on Google for your content, because at the end of the day, Google in many cases is actually incredibly inaccurate. So with this, I ended up going ahead here and I took this screenshot, and with it, I put in the phrase, the 3 domains of life. And if you ever know, there's like a little section on Google where it says, people who ask this question, ask or also ask this. And then you can pop on it and you can click it up and they'll put like a little paragraph description there. So with it, it says, please circle the term below related to the accuracy. Is it 100% accuracy or only, again, 100% inaccurate? And again, focus only on the bacteria. And then in the space provided, use complete sentences to justify your response. So again, reading through this, it says a difference between all three domains is what their cell wall contains. No, not really. A cell wall in the domain Archaea has peptoglycan. The organisms that have a cell wall in the domain Eukarya will have a cell wall made up of polysaccharides, and a cell wall in the domain Bacteria has neither peptoglycan or polysaccharides. This statement right here happens to be 100% inaccurate. And so what Google is now saying for being true is actually 100% inaccurate because, again, when you take a look at it, the Archaea domain, they do not have any peptoglycan. This is something, again, that you're going to see with the eubacteria or the bacterial domain. And so when it comes through here and it says a cell wall in the domain bacteria contains either peptoglycan or polysaccharides, that is somewhat of a true statement. Or I apologize. It would be a true statement if they took out this word here, neither, and put in either. So again, another big mistake right here. Google is telling you that the bacteria domain does not have peptoglycan or polysaccharides. And that is just not true. This word here should be either. And then again, Archaea, they don't have peptoglycan. That's something back to the, eubacteria. So this is an application kind of short answer type of question. And again, it goes back to the objectives, okay? So the reason why I'm showing you this question, just like I'm showing you question number 2, these are application questions. They're where you put thought into. Question number 1 is really rote memory. I shouldn't have to really go through many questions with number 1, okay? All right, going on to question number 4. Remember, don't worry about names of bacteria. Ask yourself, what is the question? And then go back to the question later on. So, Rhizobum lugnasorium is a gram-negative bacillus that produces lactic acid as a byproduct of fermentation. If you wanted to find these organisms in their natural environment, you would have to look in the soil and specifically around plants that are legumes. Peanuts, soybeans are examples of legumes. The bacteria are not hazardous to plants and do not cause any known diseases in humans. These bacteria grow well in the temperature around 29 degrees Celsius. Which of the following characteristics are present in this species of bacteria? So a couple of characteristics, right? I talked about gram-negative bacillus, okay? Went through that. Didn't really go ahead and talk too much about the environment. The environment's not even a characteristic here, okay? And then the other part here, again, don't have to worry about it causing disease because we're not looking at diseases. Diseases are not until exam number 4. The temperature is kind of unique here because it talks about it growing around 29 degrees Celsius, okay? So taking a look here at, again, the objectives, all right? Because, again, people ask me the question, where do these objectives come from? So, right here is a little prokaryotic cell structure here. And so with that, describe the bacterial cell wall and understand the role of the peptoglycan in its structure. Number 4, describe the outer membrane of a gram-negative bacteria and know the relationship of the lipopolysaccharides to the toxic properties of the cell wall. All right? So I kind of went up with that one. And then the other one too here happens to be that one about temperature. So if I go into the next set of objectives, which again, I have to scroll down here too. So right here happen to be the temperatures. You've got the psychrophiles, mesophiles, and thermophiles. So these all happen to be the different temperatures that, again, I had in the videos for you. So looking at, again, the practice question here, the bacteria would gram stain pink due to the presence of high levels of peptoglycan in the cell wall. Letter A is a false statement. Gram-negative do stain pink, but it's actually because they don't have a lot of peptoglycan. So remember, gram-positive, positive purple, okay? And again, that means they have a lot of peptoglycans. So this one right here, this is a false statement, all right? Since the bacteria are found in the soil, they are spore formers. You don't have to be found in the soil to be a spore former, but many bacteria that are in the soil are a spore former. There is nothing there at all about that. These bacteria are mesophiles. All of the above are true, none of the above are true. Actually, the statement in here that happens to be true again is this concept of a mesophile. Remember, mesophiles grow very good again at those temperatures that are similar to, if you will, human body temperature. So again, 29 degrees Celsius is close again within that range, and so that would be a mesophile. Okay? All right, this one right here. All right. Kind of funny, I just literally took my kid to the dentist, and he had a cavity, and FYI, while I was going through a root canal last year, I thought of this question, and I put it in here because at the end of the day, I have nothing else to think about when you go in for a root canal. So I wrote about this bacteria that in many cases, this bacteria happens to be an anaerobe, and so because of that, this is one that causes some of those really deep infections. So anyways, going forward here, so this bacteria right here, Polyromanus gingivalis, is a gram-negative bacillus, obligated anaerobic pathogenic bacteria. Some of you are going to be like, oh my gosh. But you know what? To be honest with you, you need to feel comfortable with using the terminology. Using terms like, hey, this is a gram-negative bacillus. It's anaerobic. You need to start feeling comfortable putting all that terminology because it simplifies things. It's 100% accurate. You need to get used to it. And you need to be able to apply it. Okay? So with that, this bacteria is found in the oral cavity and it causes periodontal disease as well as other diseases. The bacteria do have fimbriae, allowing them to attach to the surface of the teeth. And if you were able to grow the bacteria in broth culture, which of the following would be the test tubes that would be the most similar to the growth you would expect? Well, back to it, the big thing here about these test tubes, it's all about growth. And remember, the top of the test tube is going to have more oxygen. The bottom of the test tube is going to have less. So when you take a look here, the big important concept here influencing the growth is the fact that the bacteria is obligated, which again means it has to. Anaerobic means no oxygen. So again, if the top of the test tube has oxygen, the bottom of the test tube has no oxygen, well, again, where would you expect this growth? You would expect it here in tube number 2 because all of the growth is at the bottom. Number 1 would be if you had a obligated aerobe, where again the bacteria are growing specifically at the top. And so with that, number 3, this would be a facultative anaerobe. And then number 5 right here, this would be a microaerobe, not that you have to know what that one is. So again, these tubes over here all get eliminated. And again, the important thing here is that this is an obligated anaerobe. And again, going back here into the objectives, the oxygen, anaerobic, obligated, right? That was a characteristic. And if you even go into the PowerPoint lecture slides, you will see nearly this identical image looking back at you. All right? So with that, I really want you to slow down, ask yourself again, what are the questions here? You know, then go back and reread for the important things, okay? Don't panic about names of bacteria, species of bacteria, or anything of that nature, because again, you got to focus on the question at hand. And if you do that, you will be okay, all right? And I hope with some of these application questions I just showed you that you had a chance to see these questions and where they come from. Because like I said, I'm going to go ahead and write questions that are based off from the objectives. And I'll put them in some sort of sequence here in the exam. So that way, again, you're not having to jump around. Because again, I don't want you to be, setting yourself up for failure by having your brain go in all these different patterns, if you will. Okay. At the end of the day, again, you do have your note card there, so feel free to use that. And if you have any questions for me, feel free to ask me at some point. Send me a message, and I'll get back to you as soon as I can. All right? Otherwise, I hope you all happen to have a good day, and I hope you had a chance to take a look at this video. And I will see you at some point tomorrow, provided we'll have class. They're talking about having potentially a snowstorm tomorrow with some high winds and things of that nature. So we'll see how that stuff goes. Take a look at the weather in Texas. It's kind of weird, but I think we're actually in a better shape than what Texas is. So anyways, have a good night, stay warm, and I'll talk to you later. Bye-bye.